Nutrition of Transplantation Immunology. So it's extension to our previous discussion regarding transplantation immunology. We'll continue with the same topic of transplantation immunology, but more emphasis on transplantation tolerance and with the respect of hematopoietic stem cell transplantation. So by definition, the operational definition of transplantation tolerance is a non-reactivity to an alloantigen without compromising the activity against an antigen, other antigens or pathogens. So I would invite Dr. Chakravarti. So welcome, sir, to discuss transplantation tolerance. For the sake of all of us, better to say juniors. So I would request him to explain first about the T cell reactivity that will build up the foundation of transplantation tolerance. So now I would request Dr. Chakravarti to discuss about this. Thank you.
Thank you, Sarita. Thanks, Dheeraj and the whole team of Intas for arranging these sessions. So as you were told just now that we are going to discuss about transplantation tolerance. And the question of transplantation tolerance comes when there is an issue of intolerance. Now, we hear a lot about intolerance nowadays, right? All of us, we hear about intolerance. Intolerance to dissent, intolerance to processions, intolerance to portrayal of something which we don't like, etc. Now, you will be surprised to hear, or rather may not be surprised to hear, is that nature has made us physiologically intolerant. Okay, that's what we are in a sense. So physiologically, we are intolerant. And why is that? It is to save us from the trillions of pathogens which surround us. So we cannot afford to be tolerant to the swarming pathogens all around us. We have to protect ourselves. So that is where the innate and the adaptive immune system has developed in a way where it is inherently programmed to react to anything that's foreign. The foreign thing might be an inanimate object. The foreign thing might be an animate object like, bacteria, virus, protozoa, anything. Or it can be an antigen derived from someone else. Like, let's give you an example. Pathogens, you all understand that in order to fight bacteria, our entire immune system is built in such a way that it is sort of programmed to react against pathogens, okay? Now, with that knowledge, if we look at what happens if we give a blood transfusion to someone, if we give a whole blood from someone like a third party to a patient, or for a normal person for that matter, we give tons and tons of leukocytes, which include neutrophils, lymphocytes, eosinophils, monocytes, so on and so forth, nucleated cells. Mind you, erythroid cells are not nucleated, but the white cells are nucleated and they live. They live for a significant duration. So when these cells enter our body, means with one transfusion, one whole blood transfusion, we are giving something like 10 to the power 9 to 10 to the power 10 white blood cells. Now those white blood cells, if you look for them, maybe a day down the road, you won't find them in the patient's body. The reason is that our body has rejected them. comprehensively rejected or killed those foreign cells. Now, that is the normal function or the normal physiological function of the human body. So, I have discussed a bit about the innate immune system in my last talk, but transplantation tolerance to a large extent is a function of T cells as we understand. So, my focus today will be on T cells and the adjacent events. So let's look at how an immune response develops in a nutshell. Thanks to COVID, a lot of you have heard about all these things now. How many days it takes for antibodies to develop and how long they last, etc., etc. And even in newspapers, I see about, oh, don't worry if it doesn't respond with the antibodies, it produces a T cell response. Fantastic. So T cell, B cell antibodies, these are pretty routine lingo right now. So with that benefit, let me just take you through this very plain and simple slide, which shows that once the body is faced with a pathogen, now any pathogen or any living substance is built with proteins, amongst other things. And proteins, once they enter our body, are broken down into peptides. Now, these peptides are presented to the adaptive immune system. Means there are two parts of the immune system, innate and adaptive. The innate immune system is triggered by,
Hello?
yes, hello. Uh, the innate immune system is triggered by any or all of these peptides uh, to produce what we call inflammation. Now, inflammation is a way by which the cells which react against a pathogen would gather to the site of the infection. That is the job of inflammation. Now, too much of inflammation is not good. As we also have seen in COVID, that too much of inflammation is not good. So the innate immune system quickly transfers its job to the adaptive immune system. If this process cannot take place smoothly, then there will be abortive inflammation, means there will be a lot of inflammation without the T cells being able to do their job. The problem can be at various levels, but the major job of the innate immune system, let's take, for example, the major cells over here, the macrophages, which basically engulf the pathogen and present the peptides, which are broken down from the proteins. Say for in COVID, I mean, it's become very useful to take the example of COVID. Now you all hear the vaccine is against the spike protein of the virus. Now this S protein uh, is broken down as peptides, and a particular part of the peptide is presented by the macrophage along with the major histocompatibility antigen, which is class 1 or class 2 to CD8 or CD4 cells. Now, if the lymphocytes, I mean the T lymphocytes can recognize these antigens as a foreign antigen in conjunction with self-MHC, they will be activated. I'll come to that. And once they get activated to a sufficient extent to mount a response against the pathogen, the whole process of contraction of the immune response starts. It is essential. Otherwise, we will be in a constant state of war within ourselves. To just give you an idea that, an average human body contains something like 200 billion of T cells. And if they're all in a state of war against various pathogens which invade our body, we wouldn't survive. So it's very important for the homeostasis of the immune systems that the immune response subsides. And once the immune response subsides, there will be a few cells of both T cells and B cells which would survive as memory cells to combat the next bout of infection from the same pathogen bearing the same antigen. So, this is in a nutshell how an adaptive immune response works. But as I told you just now, that T cells execute, but the whole process of execution is masterminded by the antigen presenting cells or the monocytes and macrophages. I mean, to be precise, macrophages, dendritic cells. These are called the classical antigen presenting cells. And some of these cells, you know, are very good at antigen presentation. Some of these don't present antigen that well. Some of these are very aggressive. You know, the classical dendritic cell would be a classical, you know, antigen presenting cell like doing a Bharatnatyam. Now, the plasmacytoid DC would be something like, you know, a Salman Khan dance, which is not a dance at all, which doesn't work out. Uh, but if you look at the inflammatory DC, it's like a thunder. They would be very aggressive in their presentation. So, with the knowledge that you know, these are the these are the antigen presenting cells, and they are presenting antigens to uh, the T cells. As I told you earlier, that it all depends on the uh, how the peptide is presented to the T cells, whether this antigen presentation is to a CD4 cell or to a CD8 cell. And as I'll tell you in due course, it's not just the antigen presentation. There are multiple other factors which will dictate that how the immune system response is going to take shape. It's like giving birth to a child. The first antigen and the T cell mulakat, as you can call it, interaction is like giving birth. And after which, how it takes shape is a very interesting story. So, as we had told you in our second session in the series, that MHC class 1 and class 2 are very distinct pathways of antigen presentation. And this is important in understanding the concept of tolerance. The class 1 MHC pathway, which is HLA A, B, and C in classical terms, uh, they present antigens to CD8 cells. And these MHC class 1 present antigens in association. I mean, the antigens that they present or the peptides that they present are usually intracellular. Means to a large extent, these are viral, to make it very simple, because a virus internalizes, whereas the bacteria or even fungus, they are extracellular. So protozoa or mycobacteria or virus, they are intracellular, and they would be presented in conjunction with class 1 MHC to CD8 cells. Now, this is the same pathway through which our cancer surveillance works. Because if there is, for example, in a physiological process, a cell is old, senescence, so it will die down. Its breakdown of the intracellular proteins is going to be presented on the surface with the self-MHC, but these proteins or the peptides for that matter, which would be presented, are damaged. So they are not intact self-peptides. So these are damaged self-peptides, which would be recognized by the CD8 cells, and they would kill the cell immediately. So this happens with carcinogenesis, and this is how the cytotoxic T cells are constantly surveilling on the causation of cancer or preventing the same. And the same job is done by natural killer cells. So these are the two cytotoxic cells in our body, means they can directly attack cells. The class 2 MHC pathway, on the other hand, presents extracellular antigens or peptides. Along with the MHC class 2, it is presented to the CD4 cells. Now, while CD8 cells are, cannon-bearing soldiers who are just going to kill. Once they find something as foreign, they're going to kill. But CD4 cells orchestrate the whole things. They are more like a general in the battle. So they are going to, based on, as I said, various other factors, go down the TH1, TH2, TH3, TH17, TH21, or any of these pathways, depending on what antigen it is, what cytokines are being secreted around it, what is driving its pathway to one of those, depending on the requirement of the body. So this is um, how the activation process and takes place in conjunction with class 1 or class 2. So it's very important to understand that CD4 and CD8 have very distinct roles. Now, the job of CD4 in this case is to prime the immune system, because CD4 cells in 90% of the cases are not cytotoxic cells. They don't kill the cells themselves, but they prime the entire system towards killing of the target through CD8, through B cells, which would produce antibodies, and they even indirectly promote NK cell mediated killing. But T cell activation is such an aggressive process. It's such an aggressive process that it cannot be left alone to the antigen and the T cell receptor interaction. If the T cell receptor were to bind to an antigen or a peptide, and if that were to cause a T cell activation, believe me that there are so many trivial antigens that we face all the time at a certain propensity, that again we would be in a constant state of aggression. So there is a second check, and we call it a two-step pathway, that the T cells would only be activated if there is a co-stimulation of the T cells. Now, this is something very close to my heart, because we have worked on this pathway to sort of innovate a very different way of doing haploidentical transplant, but that would be for some other day. Here, let us talk about how these, T cell activation is coordinated by the second step of the co-stimulation. Now, there are certain antigens or receptors which are expressed on T cells inherently. These are called, they are, constitutionally present on the T cells. And there are certain other co-stimulatory receptors which are induced on the T cells. So one of those constitutively present T cell, the receptors are called CD28. Now they bind to what we call B7 group of molecules, or receptors on the antigen presenting cells. Now they are classically CD80 and CD86. So this interaction between CD80 and CD86, they would be responsible for proper activation of T cells. Okay, so these receptors on T cells binding to the CD80 and CD86 molecules. Now, mind you, the term receptor and ligand can be used interchangeably. Means who is activating whom, right? The receptor, I mean, the cell which is going to be activated, that cell would have a receptor, and the other one which is contributing to the activation through binding would have the ligand. So in case where a T cell is going to get activated, CD28 is the receptor, CD80 and 86 are the ligands for the CD28 receptor. Now, the moment this interaction takes place, the T cells are activated. Now, within 24 hours of activation of T cells, the body prepares to subdue the T cell activation in due course. So how does it do it? It starts expressing uh, a receptor, which is again an inducible receptor called CTLA4. This CTLA4 receptor binds to the same CD80 and 86 which CD28 binds to, but it binds with a greater affinity. So if a T cell receptor CD28 is bound to 8086 and T cell is in an activated state, induction of CTLA4 on T cells is going to subdue that activation by displacing CD28 from this 80 and 86 and diminishing the activation. But before that takes place, as I said, this is a constitutively expressed receptor. These are induced receptors. So a whole host of receptors, which include ICOS, which will again bind to ICOS ligand in macrophages, B cells, and other cells. Mind you, B cells are also antigen presenting cells, very potent antigen presenting cells. More classically, it's a CD40 ligand, and there are numerous such. I'm not going to enumerate them today, but just know this much, that these co-stimulatory pathways, constitutive and induced, take this forward. CD40 ligand is particularly important for activation of B cells, and the rest would work in activating the T cells further down the line as well. But CTLA4 is a primary molecule which would be then expressed to subdue the activation, and subsequently later on in the course, if the activation continues, as you all are hearing a lot about these PD1 and PDL1, and I'm pretty sure that majority of the doctors who write PD1 and PDL1 inhibitors doesn't know head or tail about what I'm talking about today. So PD1 is expressed subsequently, which binds to PDL1 or L2 on various cells. This comes further down the line when an activation is persisting. But the primary pathway is CD28 and 8086 and CTLA4 and 8086. Now, saying that, this is the timeline, right? Whatever, I mean, when we loosely could talk about activation, what is activation? Activation is basically when the receptor and ligand would bind, and this binding of the receptor and ligand will start a whole array of cell signaling. What does a cell signaling mean? I mean, there are several pathways, as you know, but broadly speaking, in immunological terms, one pathway takes it down the uh, calcium channel pathway, where calcium binding to calmodulin and so on, or through the help of calcineurin, will activate NFAT. There is a RAS pathway which works through MAP kinase, and again will go intracellularly and activate APC, and so on. There are several others pathway. NF-kappa B is one of the major cell signaling pathways for T cells. But what do they do at the end of the day? These receptor ligand binding and the cell signaling, they will ultimately result in transcription of a gene. Again, as we know, that there are some 20 to 30,000 odd genes in our body, and every cell possesses them. There are certain cells, certain genes which are active in certain cells. And the gene which is, primarily activated in all T cells in this receptor ligand binding is interleukin 2. And interleukin 2 is the master cytokine in the process of T cell activation, because it drives the entire process, and subsequently other cytokines come into the picture. But the process of you know, helping in transcription of the necessary genes involves primarily interleukin 2. And once this happens, which takes anywhere after the activation has taken place between 8 to 12 hours, there is increased expression of interleukin 2 receptors. Now, interleukin 2 receptor generally exists in an incomplete form. It will now have, it usually has alpha chain, and with addition of beta chain, it increases its affinity, but unless the gamma chain is added, it is not a full-blown interaction with the cytokine and the receptor. So once this takes place, the T cell proliferation peaks at around 2 to 3 days, and that you will understand is the basis of using post-transplant cyclophosphamide. And those who have done this, mind you, I mean, George Santos and Richard Jones, who have, and of course, Leo Loznik, I mean, they have done this beautifully. This is as thorough a science as possible. That why use post-transplant cyclophosphamide on day 3 and 4 and not on day 2? That's because of this particular reason, which you see over here, that the peak of T cell proliferation, once it meets with an antigen, peaks at day 3. So now a lot of you now know why PTCY is used on day 3 and day 4. So saying that, we now go to what does T cell activation do? And I wouldn't dwell too much on it, but this is the antithesis of tolerance. So I need to mention this. Now, whether this is graft versus host disease or a graft rejection, it's the same thing. That use of chemotherapy or radiotherapy is the primary event in instigating this process. If you were to just give donor cells in an empty bone marrow without any chemotherapy or radiotherapy, you wouldn't get this sequence of events. So this is what these are classical DAMPs and PAMPs. Once the cells are damaged, the release of ATP, the release of uric acid, and by damaging the gut, the release of LPS, such a strong, driver for TLR stimulation, is the first in the chain of events that lead on to T cell alloreactivity. So once that happens, I mean, again, as I told you, the macrophages or dendritic cells, they are at the center of this. So if we can control them, we control a lot of things downstream. And T cells are downstream in this whole reaction. It's only if the macrophages can feed them with all the information they need in terms of the peptide, in terms of the cytokines, then only the T cells will be activated to cause alloreactivity. And this is what graft versus host disease is. And look at it from the other side, that's a graft rejection. Now, the other thing that's very important and not often understood in the process of, GVHD or the process of graft rejection is that the T cells have to migrate. And there are key chemokines and chemokine receptors. So the chemokine receptors, for example, are CCR7. If you do not have CCR7 on T cells when they're maturing, they are not going to come out of the thymus and they die inside the thymus. So imagine how important it is to have the right chemokine receptor for traveling. It's a simple travel. But without those receptors, T cells cannot survive. Now, for these T cells which are given with the graft, or classically the naive T cells, they have a ligand called CD62 ligand, which drive them towards secondary lymph nodes or homing to the secondary lymph nodes. Okay, like CXCR4 expression drive stem cells to go to the marrow, okay, where they will bind to CXCL12. So here the 62 ligand will drive the T cells to secondary lymph nodes, and without that, a process of GVHD cannot be initiated. Of course, there are several other receptors which come in at various stages of this alloreactivity, but I just give you a broad picture of how this is such key to um, the T cell alloreactivity. So, if you look at it in a broader perspective, so the cytokines which are there as a result of inflammation, largely produced by the macrophages, would drive the T cells towards one or the other pathway, Th1, 2, 3, 17, or regulatory, or 21. And this happens through transcription of certain genes, which are called the master transcription regulators. And these cells, how do we identify these cells? We are not always going to do the gene expression, because these cells produce signature cytokines, which I'll just show you. So take, for example, Th1 pathway. The dendritic cells or the APCs are, presenting the foreign antigens or pathogens to a naive T cell. Now, if this is LPS pathway, they are going the monos. I mean, mind you, the monocytes and macrophages also are directed towards one pathway or the other. So whatever T cells are producing, the macrophages are also capable of producing similar cytokines. So the most of the times with a gram-negative pathogen, or for in a alloantigen situation, IL-12 is going to be produced. Now, the IL-12 is going to drive the T cells down the Th1 pathway. So the signature cytokine, the polarizing cytokine is IL-12 and interferon gamma. Now, these are produced also by NK cells. And again, I'm not getting into NK cells today because it's a completely different, discussion. So I'm keeping NK cells temporarily out of the picture, just focus on the APCs and T cells. Now, once that happens, there will be induction of STAT1 and STAT4, okay, in the JAK-STAT pathway. That's again a very key pathway, the cell signaling pathway, which results in transcription of T-bet. Now, what happens following this? These cells, what we are now used to calling Th1 cells, will be producing interferon. And what is interferon going to do? Interferon is going to drive the NK cells and the CD8 cells to potentially cytotoxic pathway, and it's a feedback pathway also. So this will influence the APCs to continue this process in a vicious cycle, unless the T cell activation is stopped by the inhibitory co-stimulatory receptors. And in a similar way, the Th2 pathway, which is classically told as a helminthic pathway, is driven by the polarizing cytokine IL-4, where there is through STAT6, there is induction of GATA3, which is the equivalent of T-bet for Th2. And here the polarizing cytokines are IL-4 and IL-13. And now this IL-4 and IL-13 will take the macrophages or dendritic cells down to what we call a M2 phenotype. So again, it's a vicious loop where they keep on feeding each other. And the third pathway, the third inflammatory pathway, is what we have known quite late in our, knowledge of immunology, is called the IL-17 pathway. Now, this is driven by IL-6 and TGF beta. And it goes through the STAT3 pathway and induces what is called a ROR, or ROR gamma T gene. And what does it do? It produces IL-17 and IL-22. And this is supposed to be an autoimmune or neutrophil recruitment pathway. So this is what the concept is of. These are the antigen presenting cells. These are the polarizing cytokines, and these are the master regulator transcription factors, and they go on to produce the signature cytokines from each um, you know type of T cells in the different pathways, which leads to a different effect. Like this is a pathway which will drive the B cells. This will drive the CD8 cells, and this will drive the neutrophil macrophages, etc. So if that part is clear, then I would take a bit of break
Yeah,
because
thank you, sir, for explaining so many complicated pathway in a very simplified way. So hope we have been able to understand this. But after such a elaborative discussion, what comes in the mind is how the self-tolerance develop. So can you tell us a bit about the self-tolerance, establishment of this tolerance, how it happens inside our body?
this is really a fascinating thing. I mean, mind you that when I studied medicine in the early 80s, Um, one of the things I did was read the textbook of Harrison inside out maybe 100 times. And I wish that, we had more knowledge of immunology at that time. Then I would have been, without any exaggeration, one of the pioneers in immunology by now. But unfortunately, we had very little knowledge other than CD4 and CD8 cells at that time. And you will be surprised to know that majority of what we are talking about today is a knowledge which is gathered over the last 2 decades. And to keep up with the developments in immunology, which happened like an explosion in the last 2 decades, is mighty difficult. And what we are essentially doing is we are trying to understand the nuances of nature. Mind you, we are not developing anything. We are not developing anything. We are trying to understand. We are trying to understand how nature works. And if we can understand it properly and apply it properly, then only we can succeed. And these are all experiments which have taken place maybe in the 70s and 80s in the lab by some great people who have worked for 2, 3 decades on the small things. And today by a drop of a hat you can cut a gene, like gene editing. But mind you that in those days they worked with such small tools. Okay, even labeling a cell was such a difficult task. And these experiments on mice, which have established what I'm going to talk about now, is a phenomenal advancement in immunology, although we don't know a lot of it still. So the issue is that when a T cell develops, why doesn't it attack its own body? And just imagine what nature has done. I mean, such fine tuning is not humanly possible. So, how is it possible that the same T cell, which is acting against an antigen, which is so closely related to the self, mind you that HLA-A2 with an A2 antigen with O2 allele, when it faces an HLA-A2 antigen O3 allele, it reacts. Not to O2, but O3 allele, which is differentiated by a couple of peptides. It reacts against single nucleotide polymorphic sites. How is it possible? I mean, this has intrigued me. That's why I'm talking like this, because this is what is the basis of T cell immunology, you know. So this is a sort of schematic diagram, and I would request you to look from the top. So once a T cell, I mean, once a common lymphoid cell is formed in the bone marrow, the ones which would go migrate to the thymus would have expression of CD44 antigen. Again, look at this. Such an important receptor just makes it go to the thymus with expression of c-KIT. I don't know if everything is written over here, which I don't think it is, but just I rarely, I'm able to do slides and read out from slides. So just try to listen and understand what I'm saying. So these cells, the common lymphoid progenitors, will express CD44 and c-KIT. Very high expression of c-KIT. c-KIT is CD117, which you use in diagnosing leukemias. It just is a primitive a marker of primitivity. So when these cells enter the thymus, they enter into the thymic cortex. Now, once they enter into the thymic cortex, they just first express the CD3 receptor. That also is a cytoplasmic CD3 receptor. So they are called a double negative cell. And these double negative cells, without complicating it much, I would say that they would go through 4 steps, which are called DN1, DN2, DN3, DN4 in the cortex. And the first receptors which are expressed in the fetus are 3, which are gamma, delta, and beta. Now, gamma and delta are the first receptors to appear. And if the gamma delta receptors are borne by the T cells, they are gamma delta T cells. But our body has majorly alpha beta T cells. So how it happens? So these, as I said, there are 3 receptors which are transcribed at that stage: gamma, delta, and beta. Now the next step is called beta selection. The cells which will undergo beta selection means those who will express beta receptor will lose their alpha beta, will lose their gamma delta receptors and will go down the alpha beta pathway, which happens to 95% of the cells. And 5% of the cells which will not undergo beta selection will remain as gamma delta T cells, and they leave it from the cortex. So during this maturation pathway in the cortex, um, these cells, double negative cells, gradually lose the expression of 117. This will help you understand also that when you are diagnosing T cell ALL, why is expression of certain receptors at certain stages is important. And the same, they will gradually lose CD44, and they will gain CD25 and then again lose it. But the gamma delta will have the CD25 receptor, and they will exit the thymus at that stage. But the ones which have undergone beta selection will be in the thymus. Now, in the thymic cortex, there is a very interesting process that takes place, okay? That the thymic cortical epithelial cells would present um, the developing T cells with self peptides along with self MHC. The T cells, while they're expressing their beta receptor, gradually express the alpha receptor as well. And there is a germline huge amount of recombination in the V, D, and J segments of the receptors at a germline level. So there are some, 1 billion combinations possible. And each of these T cells are presented the self MHC with self peptide to by the thymic cortical epithelium to the T cells. It's like a queue, a queue which goes on one cell at a time. And any cell which would recognize the self MHC with self peptide with high affinity would be killed or energized. Means they would not be functional T cells. The cells which identify self MHC with self peptide with a much lesser affinity would be allowed out of the gate. Now you can go to the thymic medulla. So this is the first step. Now, when you are going from thymic epithelial to thymic medulla, what is very interesting, and that's why I've used this picture, is that look at this, that what happens to 90 to 96% of lymphocytes which develop, they just don't interact with the MHC. And what happens to these cells? This is called death by neglect. Or in other terms, if it happens in the periphery, it's called death by ignorance. So very, fascinating way of regulating autoreactivity by the greatest dangers. Means these are like children. If they are nurtured properly, they would be well-behaved children, which will take care of the parents. And if they're not, they would grow up to ruin their own parents. So exactly that's how the thymic epithelium develops. So only 2 to 5% of the cells would go on to develop as mature T cells and will go into thymus. And 2 to 5% would again be energized. It's called clonal deletion, or they will undergo energy because they have identified the self MHC with high affinity. Now, what happens once they enter the medulla? And that is what I think is the marvel of nature. I mean, it's unthinkable. I mean, if you ask me, those who discovered it, they are the greatest scientists. And to talk about it, I feel sort of choked to talk about it. Just tell me something that there, in cortex, the thymic epithelial peptides were presented with the self MHC, right? But the pancreas, the brain, the lungs, the kidneys, they don't all express the same peptides. So once these cells go out and they face all these different peptides originating from different organs with the self MHC, they are going to react. So how do I stop that? There is a gene called autoimmune regulator, AIRE. That's the secret. That's expressed in thymic medulla. If it is not, there is humongous autoimmunity. So what they do, as I said, that we are capable of expressing 30,000 genes in every cell, but we don't. So medullary epithelium expresses AIRE, autoimmune regulator, which actually is capable, mind you, it's capable of producing peptides representing every single organ of human body, and it samples every T cell, which is basically 2 to 5% of which they have entered, with a specimen of peptide from every single organ. It's only after they have gone through this test of AIRE that they are allowed to get into the circulation. Isn't it fascinating? Now, if they don't, they die. They die by apoptosis. And the dendritic cells and macrophages in medulla also got a huge part in presenting this uh, peptides, cell peptides from different organs, and also to engulf the apoptotic cells. But what happens over here is there is another critical pathway, that is the ability of these cells to express CCR7. So if they can express CCR7, they go through the scrutiny of AIRE, they will leave the thymus as mature T lymphocytes. Another pathway which I'll come to, I mean, another trick is there which I'll come to in a little while. But this is like an NDA, National Defense Academy, where you have to go through the grilling for 4 years, or MBBS from or MD from PGI Pankaj would recollect. And that when we went in, we didn't know that we would get out of it. So this is something I've developed, I have described right now. Now, the other part is, how do we decide which T cells would become CD4 and which T cells would become CD8? So these are fundamental questions which immunologists, as I said, in the 70s and 80s, they have toiled to discover. And there are several pathways which have been discussed and put forward as how it happens. But in a nutshell, what I'll tell you, what I believe is the most likely happening is that, see, nature always, deals with things with the thing in mind that if something goes wrong, I have a backup. So when a T cell is, I mean, is receiving a signal in the when the T cell is receiving a signal in the thymic cortex and the thymic medulla. Now, the continuous signaling does not give rise to CD8. It gives rise to CD4. But interrupted signaling would give rise to CD8. Why is that? The reason is that CD8 cells are cytotoxic cells. They kill. They don't think anything before or after. They are born to kill, they will kill. Arnold Schwarzenegger, it's going to kill. CD4 cells are the guiding cells. So they know how to handle signals. So interrupted signaling will give rise to CD8, and continuous signaling will give rise to CD4. That is how these cells are primed at the outset. And that is why you have more CD4 cells than CD8 cells. You don't need killers roaming around, isn't it? So once the T cells are out in the open, that's out in the circulation, have we achieved a perfect state of self-tolerance? The answer is no. We still can get autoreactivity. And if this autoreactivity goes unchecked, it results in autoimmunity. So the understanding of autoimmunity or autoreactivity and how it's controlled will help in understanding the concept of transplantation tolerance. So I have already described how a normal T cell response works. But some cells in the thymic medulla would be energized. Some cells in the thymic cortex would be energized. They would not be deleted. They would not be killed. Now, those cells in the thymus express a transcription regulating gene called FOXP3. Now, expression of FOXP3 in those T cells, which are CD4, largely. So the expression of FOXP3 in those CD4 cells endows them with a unique property of regulating the T cell mediated inflammation or controlling the T cell mediated inflammation. So this happens in thymus. We call them regulatory T cells. We call them thymic regulatory T cells. Now, when there is a antigen and antibody, when an antigen is presented to T cells in the periphery, but the T cell, it's presented at such a low threshold that the T cells don't at all react to that antigen, even after being bound. These T cells die because of ignorance. We called in thymus death by neglect. Here we call it death by ignorance. And this happens to a large extent when, even after coming out of medulla, if some T cells retain, have not gone through the scrutiny or have escaped the scrutiny when they're presented very tiny amounts of, say, peptides from pancreas, and they just bind to it without producing any reactivity, they become energized. It can happen to anything. The same thing can happen to a foreign antigen. And development of energy is one key step in development of tolerance. The second important step is suppression. I just described that regulatory T cells are produced in the thymus, but regulatory T cells can also be induced in the periphery, which I'll show you subsequently. And these cells control the immune reactivity, whether it's autoimmune or an alloimmune reactivity. The third is that because of a futile binding of T cell with the antigen, the T cells are clonally deleted, means that clone of T cells are deleted, full stop. And the fourth is called exhaustion. Now, for exhaustion to happen, you need continuous presence of the antigen. The classical example of exhaustion is HIV infection or hepatitis C virus infection, where the inability to clear the virus, the retention of the virus or the antigen at a low level leads to exhaustion of T cells. And when the T cells are exhausted, they express PD-1. PDL1 is expressed by the corresponding cells which harbor the virus, or a cancer when you're talking of, T cell exhaustion in cancer. So this is a pathway which is not a primary pathway of tolerance, but it is a pathway of immune escape for uh, pathogens or malignancies. So if I could have made it clear that energy and suppression would lead to something called an adaptive tolerance. Okay, so this is an acquired adaptive tolerance. Transplantation tolerance is not inherited, or for that matter, no tolerance is inherited. So this is an adaptive tolerance. Get this term clear. It's an adaptive tolerance which happens through the antigen and the T cell receptor interaction without the co-stimulation taking place, which renders the T cells energetic, or the T cells are faced with such low amount of antigens that they don't react. So they're eliminated by ignorance, due to ignorance. And of course, the major pathway also is the regulatory cells, which I'll show you briefly, which control this process of alloreactivity. And as I told you earlier, that IL-2 is the main driver for T cell proliferation. But what IL-2, how the body has regulated the whole thing is that once the T cells, the effector T cells, again, the terms are important, what we call T effector cells, and the other cells I would call T regulatory cells or TREGs. So T effector cells, or the other term which makes it easier to say is conventional T cells or T-cons. So let's say T-cons or TFs. So T-cons do not express IL-2 receptor constitutively. Once they're activated, they increase the IL-2 receptor expression. And because of IL-2, they get activated, they proliferate, etc. On the other hand, the regulatory T cells, the IL-2 receptor alpha is CD25. So CD25 is expressed constitutively on gamma delta T cells and also on regulatory T cells. So IL-2, if you have in limited amount, they are going to be taken more by regulatory T cells than the affected T cells or T cons. So this is the sort of fine tuning of regulation that takes place. Okay, so that within a given pool of IL-2, um, once a T cell is proliferating, okay, I give them the IL-2. But once I see that they're going beyond control, the regulatory T cells are there to mop up the IL-2 so that they don't proliferate. So that's one of the mechanisms by which the regulatory T cells control the T cons or the affected T cells. And I've already discussed this co-stimulation issue. So um, I would, you know, uh, just summarize this part by saying that T cells are the actors. I mean, this is the best way to visualize and remember and understand it. So they're directed by the APCs, how they would react, Th1, Th2, Th17, T regulatory, blah, blah. So APCs are the directors, but APCs can't do anything unless the money flows. So the directors cannot direct the actors to behave in a particular way or to act in a particular way unless the money flows from the producer. And who's producing the money? The cytokines. The cytokines secreted by APCs and also the T cells themselves. And, you know, how you go from one set to the other, how you can smoothly execute the whole T cell reactivity without causing problems, it carried out by the chemokines and the chemokine receptors. So that would be, in a nutshell, what is alloreactivity and how tolerance develops, both self-tolerance in the thymus periphery and allogenic tolerance, which can develop, which one, which might develop.
Thank you, sir, for this explanation in very simple way. So from your discussion, we understood that the several cellular component and as well as some soluble components are important to develop the establishment rather of this transplantation tolerance. So nature has, you have rightly said, the nature has really geared our body in such a way, and such complicated processes are going inside the body. So hence, I would request you for sir to explain a little bit of this cellular component first, and later we'll discuss the soluble component.
As I have already told you during the course of the previous discussion, that the key to tolerance are the regulatory T cells. At least that's what we know sitting in 2021. I'm pretty sure in 2031 we'll know something different, because the knowledge is evolving, and unless we are ready to accept the evolution of knowledge, we cannot progress. I mean, what I find is, what I standing today, what I find is the difference between the ancient Indian education and the current Western education. When I talk of ancient Indian education, don't mistake me as some bhakt or something, please, because we are too rich culturally and we know very little of our heritage and tradition. The opening prayer of Vedas used to be that the nature is supreme, the nature is God. We bow down to the nature and we bow down to learn from you. In the Western education, unfortunately, the paradigm is what I do not know does not exist. It was directly the opposite 10,000 years ago. And the amount of knowledge gathered at that time was unparalleled in every sphere. And that's why I mean, unfortunately, I am in no formal teaching system. But whenever I get an opportunity, I tell the people that humility is the core to attainment of knowledge. That unless you accept the limitations of human beings, you cannot learn, you cannot know more than what is provided to you through books, because it's all there in nature. It's for us to discover. With the attitude that what I do not know does not exist, does not lead us anywhere. So today I might say that regulatory T cells are the be all and end all of tolerance. It might not be the case because we do not know a lot about what other things are going on. We know a bit about regulatory T cells, as I said, in the last 2 decades. And as I described to you, these are the cells which develop in the thymus, which come to the periphery and regulate the activation of T cells, effector T cells, in a measured way. Now, as I told you in thymus, when the T cells recognize the self MHC with self peptides with high affinity, they are clonally deleted. If they recognize with much lower affinity, several folds lower, then they are allowed to go through. If they recognize with an intermediate affinity, they are the ones which go on to become regulatory T cells. And the primary event in development, I mean, in a T cell becoming a regulatory T cell is expression of FOXP3 in these cells. Now, what happens in these cells is that they are constitutively produced interleukin 2 alpha receptor. They constitutively produce CTLA4. And when these cells bind to a dendritic cell, they would sort of mellow down that dendritic cell or any antigen producing cell for that matter. But how it exactly works is again $1 million question. And mind you, in 2 decades, we are not sure how it works. One mechanism is, as I told you earlier, is by uh, depleting IL-2 for effector T cells. But what I think is more interesting and a more valid way of its functioning is that these cells produce IL-10 and TGF beta. And IL-10 and TGF beta, as I'll tell you subsequently, are the key drivers of immune tolerance or immune regulation. So TREGs or T regulatory cells um, would not work unless they also are presented with the antigen. So like T effector cells need antigen presentation, regulatory T cells also require antigen presentation to be active and to do the job. So the same antigen which is presented to the effector T cells would be presented to the regulatory T cells, then only this whole phenomenon can happen. Now, the other very important cell which regulates the whole cascade are the macrophages. Um, I mean, I would not go into, this is a very complicated slide by the looks of it. But what, if you look at it carefully, that macrophages are the ones which first encounter the antigen, process the antigen, produce cytokines, drive T cells down certain pathways. So classically, the M1 pathway or the which corresponds to the Th1 pathway of T cells, they happen by the macrophages producing interleukin 12. And these macrophages would drive T cells down Th1 pathway. But if they do not produce IL-12, they produce IL-4, they would drive it down the Th2 pathway, and these are M2 macrophages. Now, within these M2 macrophages, there are several subtypes which have been talked about and recognized. So they have got several functions. I'm not going into details as M2A, B, C, or D because I personally don't adhere to this nomenclature strictly. But I would say that there are three broadly. There are rather four types of macrophages. The fourth type I'll come later. The first one is M1 macrophages, which drive T cells down the Th1 pathway. The M2 pathway, which the major M2 pathway would drive it down the Th2 pathway. Now, there are certain elements of this M2 pathway which would also lead on to, fibrosis and wound healing, etc. And these are what we call the regulatory macrophages, which predominantly, like T regulatory cells, secrete IL-10 and TGF-beta. Secretion of IL-10 and TGF-beta drives the um, cytokine, drives the inflammation milieu down the uh, regulatory pathway. And fourth, again, are the tumor-associated macrophages, which are somewhere a mixture of they have a mixture of M1, M2, and regulatory in the varying composition. And it is often believed that they have more of M2. That's why they help in tumorigenesis. But if they have more of one M1, they might not. But it's not that straightforward. But for the sake of transplantation tolerance, eh, the regulatory macrophages which secrete IL-10 and TGF-beta are key for that. Now, as I told you earlier, that Tregs are not the only cells. There are several other regulatory subsets which are recognized in very small amounts in circulation. And they all are capable of producing IL-10 and TGF-beta. They are either called TR1 or TH3. There are CD8 tregs, which also express FOXP3. There are regulatory B cells, um, which produce IL-10. There are myeloid-derived suppressor cells and mesenchymal stromal cells, to name a few. I'm not bringing NK cells intentionally into this, but NK cells and NK T cells also have a very dominant role in establishing tolerance. So I will next go to these two major cytokines, um, which are IL-10 and TGF-beta. Now, if you look at IL-10, they are produced by all these regulatory T cells. And production of IL-10 drives the T cell down the regulatory pathway. So when we say that a particular T cell, a CD4 T cell, which is not lineage assigned yet, in presence of IL-10 and TGF-beta would become induced or peripheral regulatory T cells. So in presence of macrophages which produce IL-10 and TGF-beta, CD4 T cells which are not lineage committed would become regulatory T cells and control the T cell reactivity. And in presence of TGF-beta, if there is IL-6, it can go down the Th17 pathway. But if there is if along with TGF-beta, you have IL-2, it will take you down the regulatory T cell pathway. So this is how this is how the whole thing is fine-tuned. If you look at the TGF-beta, for example, mind you, both IL-10 and TGF-beta, they both are implicated in the pathogenesis of malignancy. And this is largely because these cytokines propel the immune reactivity towards a regulatory pathway or blunts the T cell reactivity down Th1 pathway. So TGF beta to a large extent would prevent the apoptosis of the regulatory T cells, uh, whether it's in the thymus or in the periphery. And this combination of IL-10 and TGF beta is primarily the polarizing or governing cytokines which drive the T cells towards tolerance. But in the whole process, there are there is again a very interesting phenomenon which takes place in every cell, and something again which has been understood in the last few years is that the ATP pathway. Now, every cell has got ATP. It's needed for its survival, for its respiratory cycle. It produces a certain number of ATPs for energy. Now, when these ATPs are extracellular, they are inflammogens, means they drive the T cell activation or macrophage activation pathway. ATPs are PAMPs, which would stimulate TLRs, drive down T cell activation. So, what happens? If ATPs are produced in abundance extracellularly, and how can it be produced? It can be produced when there is leakage from cells, and how can it leak from cells when the cells are damaged? So, the cells can be damaged because of, in our context, in hematopoietic cell transplantation, when we have conditioned the patient, when we have killed the cells, there will be humongous amount of ATP released along with uric acid. Now, when these humongous amount of extracellular ATP is there, it causes a mayhem situation of driving macrophages down the M1 pathway through the TLR ligands and subsequent activation of T cells. The body has got a mechanism by which it controls this. Now there are two enzymes which are expressed on surface of T cells and predominantly on regulatory T cells, which are CD39 and CD73. They are called ectonucleotidases. What they do is break down ATP to adenosine by two-step pathway. First CD39, then CD73. So you need both these enzymes, CD39 and 73, to break down ATP to adenosine. And adenosine will bind to adenosine receptors. Now there are two types of purinergic receptors, P1 and P2. The P1 are the adenosine receptors, P2 are the P2Y and P2X receptor. They're all these are ATP receptors, these are adenosine receptors. So once adenosine, the ATP is converted to adenosine, the adenosine will bind to A2AR receptor in APCs as well as the T cells and subdue the T cell reactivity. Okay, and of course there are other A2 receptors which don't do the same, but I'm not going to the details. Because for every have there is not there is a have not in science as we understand it. But I'm trying to make it simple that ATPs are inflammogens and adenosines are regulators. So more adenosine is better for regulation or tolerance. More ATP is bad for that. So again, bringing you back to the concept that if you have more, tissue damage from conditioning and produce more ATP, there is going to be more T cell activation. So saying that, I would, summarize by saying that the development of tolerance depends on the cytokine value, as I told you, that it's the interplay between IL-2, IL-10, TGF-beta, IL-6, and all IL-12, and all these cytokines. How are they produced? When are they produced? In what amount are they produced? Because mind you, these cytokines are very short-lived, and although they can act in a sort of, a lot of them work in autocrine and paracrine manner, they can work in an endocrine manner as well. But because of their short half-lives, they act on adjacent cells. So the cells which would interact have to be in close proximity to each other, and that's why the chemokines and the chemokine receptors play a part, that to bring the cells together. And antigen presentation, how the antigen is presented, in how much antigen is presented, in what milieu the antigen is presented. If a same antigen is presented in the presence of IL-12 and IL-33 and interferon gamma, it will drive the T cells down the Th1 pathway or the Th effector pathway. If the same happens in the presence of IL-10 and TGF-beta, they would induce a regulatory phenotype in the T cells. So it's a balance between the effector and regulatory cells which would dictate whether you develop alloreactivity or you develop tolerance.